Hello friends and welcome to my channel Easy Life Science. Here I am talking about a lac operon. So please subscribe my channel Easy Life Science. Like my video, share it to your friends and press the bell icon so you'll get the notification of my newly uploaded video. So let's start. Introduction of a operon. Operon is a operating unit which can be defined as a cluster of genes located together on a chromosome and transcribed together. Or we can say that it is a group of closely linked structural genes which are under a control of one promoter gene which regulates a metabolic activity. And this concept of a operon gave us by the scientists Francois Jacob and Jacques Monod. These two scientists were working on a Pasteur Institute of Paris. The bacterial DNA or a prokaryotic DNA is a polycystonic. Meaning of this word is that the multiple genes are under control of one promoter. But in case of eukaryotic organism, DNA is a monocystonic. Each gene has its own promoter. The lac operon is designated as a lactose operon. The lac operon codes for enzymes involved in a catabolism of a lactose. The lactose is a disaccharide which is made up of glucose and galactose. The lac operon is an inducible operon. To have a switch on this operon, there must be a need of its inducer, that is a lactose. So it is an inducible operon. Here is a diagrammatic representation for a lac operon. The lac operon shows the three structural genes lac Z, lac Y and lac A. They forms a mRNA or a single mRNA, and this mRNA get translated into a three different kinds of enzymes which are useful for a lactose metabolism. The lac Z gene coded for a beta galactosidase. The beta galactosidase enzyme cleaves the lactose molecule into a glucose and galactose. The lac Y, this gene coded for an enzyme permease. This enzyme or a protein is the membrane channel protein. This membrane channel protein uptake the lactose inside the cell, and the lac A coded for a enzyme transacetylase, and its function is not clear still. So these three proteins are translated from the single mRNA. So this mRNA is a polycystonic mRNA, and this regulation is controlled by a one single promoter, and this P is a promoter for these three structural genes. These three structural genes are controlled by a promoter. The operator, that is a O, here the lac repressor get binds. The lac repressor, it is a inhibitor protein, and this inhibitor protein is formed all the times. Normally when there is a absence of a inducer lactose, the lac I, that is a lac inhibitor gene, get transcribed into a mRNA and translated into a protein, that is a lac inhibitor protein. This is a repressor, and this repressor protein binds to a operator region. Because of this binding of a repressor protein, there is no any access to binding the RNA polymerase to a P or a promoter region, so no transcription occurs. Here is another model for a lac operon. The lac Z gene coded for a beta galactosidase lac Y for a permease, and lac A for a transacetylase. They are translated from a single mRNA, that is a single lac mRNA, and they are controlled by a one promoter shown by a P later, O for a operator, where the repressor protein binds to the operator region or a O portion. There is a binding of a repressor, and a repressor is formed all the time. For a repressor, there is another gene, that is a lac I gene. This is a lac I gene. When this lac I gene is transcribed, then a lac I mRNA is formed, and this lac I mRNA forms a repressor proteins, and this repressor protein bind to a operator, and they does not allow to bind RNA polymerase to a promoter region of the operon. So normally this operon is turned off as the repressor binds to operator, and though there is no any access to binding a RNA polymerase to a promoter region of the operon. Here are some elements in a lac operon. The operator or a lac O. This operator or a lac O, it is a binding site for a repressor protein. Promoter, it is shown by a letter P, that is a lac P, and this is a site for binding for a RNA polymerase. Repressor, that is this repressor protein is coded by a gene, that is a lac I gene, and this repressor protein binds to a lac O, so that it does not allow the access for a RNA polymerase binding for a promoter region. Then lac I, this controls a production of a repressor protein or a inhibitor protein. We can say that the lac operon is under a positive control and it is under negative control. When glucose is available, glucose is a simple sugar. The bacteria loves glucose most rather than a lactose, because it is a simple sugar and bacteria prefer this sugar first. So when glucose is available, the genes that participate in a metabolism of other sugars are repressed, and this phenomenon is known as a catabolic repression. When glucose is there, the operon is turned off because bacteria loves this glucose most. This is a preferable sugar, and there is no need to on the lac operon. So this is known as a catabolic repression by a glucose. Catabolic repression is type of positive control of a lac operon, and this is because of no formation of a cyclic AMP. This cyclic AMP is formed by a enzyme adenylate cyclase, and there is an inverse relationship in between glucose and cyclic AMP. There is an inverse relationship between glucose and a cyclic AMP, as in a medium where the cells are growing, the concentration of glucose is high, then there is a low concentration of a cyclic AMP. When a concentration of glucose is very low or absence, then a concentration of cyclic AMP is very high. This is an inverse relationship between glucose and a cyclic AMP. The catabolic activator protein, that is a CAP protein, binds with a cyclic AMP and forms a complex in between cyclic AMP and CAF, or we can say that CAP cyclic AMP complex, and this complex binds to the somewhat upstream of a promoter region and stimulate a binding of a RNA polymerase, and this will happen when there is a less glucose or no glucose. No glucose or a less glucose concentration increase amount of cyclic AMP, and increase a formation of this complex in between cap and cyclic AMP, and it binds to the upstream of a promoter region, and this region is known as a cap binding site. To the cap binding site, this complex is bind and it allow to sit the RNA polymerase on a promoter region and start the transcription. The cellular level of a cyclic AMP is controlled by a glucose because there is an inverse relationship between glucose and a cyclic AMP. Glucose is high, cyclic AMP would be low. Glucose is low, cyclic AMP would be high. Means this is an inverse relationship between the cyclic AMP and glucose. When glucose is depleted or when it is scarce, then there is a high level of cyclic AMP and formation of a cyclic AMP cap complex would be high. It will bind to a cap binding site and allow a RNA polymerase sit on a promoter region and start a transcription of a structured genes. Then the operon is under negative control. As said earlier, the lac repressor coded by a lac I gene. This is a protein. Lac repressor is a protein coded from a gene that is a lac I gene, and all the time these genes are transcribed and translated into a protein that is a repressor protein, and this protein binds to an operator, and because of this binding of a lac repressor protein, the DNA sequence get covered and there is no any access for a binding of a RNA polymerase and no any transcription, no translation, no proteins formed like beta galactosidase, permease and transacetylase. Therefore, when the repressor is bound to an operator, RNA polymerase cannot bind to a promoter and transcription cannot occur. Lac operon said to be under negative control. A lac operon, it is an inducible operon and normally it is off. It is an inducible operon when there is an absence of their inducer. For lac operon, the inducer is a lactose sugar. When in a medium where the cells are growing, there is an absence of lactose, the lac I gene is expressed all the times and forms a lac repressor protein, and this lac repressor protein binds to an operator, and there is no any chance to move the RNA polymerase further downstream of these genes, and these structural genes are not transcribed because no inducer is there. Lac I is expressed and forming a repressor protein or a inhibitor proteins. These are binding to a operator region, and RNA polymerase does not move and does not carry out a transcription and a translation. So this condition or this scenario is at time of absence of a lactose. When there is a presence of lactose is a inducer. When there is a presence of a lactose in a medium where the cells are growing, lactose or some part of the lactose is converted into a allolactose. It is an inducer, and this allolactose get binds with a repressor protein and changes its configuration. They changes the configuration of a repressor protein, and so that altered repressor protein does not bind to the operator, and the RNA polymerase carry its role. It carry out a transcription of all the structural genes, that is a lac Z, lac Y and lac A, beta galactosidase, permease and transacetylase. These proteins are formed when the lactose is present in a medium. This lactose get formed into a allolactose. This allolactose is a inducer. It get binds to a lac repressor protein and changes their configuration so that it does not bind with the operator, and RNA polymerase carry out a transcription of all the structural genes. Means these are the scenario when lactose is absent and lactose is present. When lactose is absent, the operon is off. Lactose is present, operon is on. Then the positive versus negative gene regulation. Both the tryptophan and a lactose operon involves a negative control of a genes because the operons are switched off by an active form of a repressor proteins. Because whenever in a medium there is an absence of their inducer, all the time the repressor proteins are formed and these repressor proteins are inhibiting the action of RNA polymerase, so the operon is normally off. Some operons are subject to a positive control. An activator protein is required to start a transcription. That is, for example, catabolite activator protein, that is a CAP protein. Positive gene regulation. As said earlier, the Escherichia coli or a bacteria likes a monosaccharide most. Glucose is a preferred food. The E. coli loves a glucose, and when in a medium glucose is present, the organism does not allow to turn on a lac operon because glucose is there. Organism will utilize a glucose first. After depletion of a glucose, organism will try to take up the lactose. When glucose is depleted or when it is scarce, the lac operon is activated by a binding of a cap because when glucose is absent and lactose is there, the absence of glucose increases a level of a cyclic AMP, and this cyclic AMP binds with a CAF, that is a catabolic activator protein, and forms a complex in between cyclic AMP and a CAP. And this complex, this active CAP complex binds to a stream of a promoter, and because of this binding, this complex to a promoter, it gives a strong positive signal to sit the RNA polymerase and carry out a transcription. This active form of CAP helps to RNA polymerase bind to a promoter, and so the transcription starts and translation would be there and operon would be on. When glucose is abundant, the cyclic AMP level or a concentration would be low, as there is a inverse relationship between glucose and cyclic AMP. And if the cyclic AMP is no, then there is no any binding of the cyclic AMP to a cap. Inactive cap would be there. Such inactive cap does not get bind to a cap binding site, and it prevents RNA polymerase from binding to a promoter, and there is no any transcription, the operon would get off. The DNA binding proteins can be either repressor or activators, and it depends on how they interact with the RNA polymerase. They act as an activator or a repressor. This configuration of activator proteins helps to binding the RNA polymerase, whereas the repressor proteins, this configuration blocks RNA polymerase for binding to the promoter. Here, a dual control of a lac operon. The four scenarios are there when glucose is absent and lactose is present, and these are two strong signals. Glucose is absent and lactose is present. These two are two positive strong signals for operon on. Here is a cap binding site, a promoter region where a RNA polymerase binds. This is a operator shown by a green coloration, and these are structural genes. cap binding site, promoter region where RNA polymerase binds, operator O, and structural genes. The four scenarios are there on the basis of presence or absence of glucose and lactose. First, if a glucose and lactose is there, at this time operon is off, because glucose is there, no cyclic AMP is there, no cyclic AMP cap complex would be bind to a cap binding site. It is a negative signal, so operon is off. The next scenario, glucose is there in a medium but lactose is absent. As glucose is there, no cyclic AMP, no cyclic AMP cap complex, and no cyclic AMP cap complex binding to a cap binding site, and repressor is forming all the time. As lactose is absent, this repressor would bind to a operator region. So these are two strong negative signals for the operon. Then third scenario, no glucose and no lactose. No glucose, high level of cyclic AMP cap complex would form, and this complex would bind to a cap binding site, but there is no any inducer like allolactose. So at this time operon would off because repressor are active. The fourth scenario, glucose is absent and lactose is present. These two are strong signals by which operon get on. Because no glucose, high level of a cyclic AMP, this cyclic AMP forms a complex to a catabolic activator protein known as a cap cyclic AMP complex, and this complex would bind to a cap binding site. This is a first positive signal, and lactose is there because lactose is the inducer for a lac operon. RNA polymerase would sit on a promoter and start a transcription. So the fourth scenario, operon would active, it get on. Here is another schematic representation, but in the text box one question is asked: what happens if there is a deletion of some genes on lac operon? If the operator is negative, that means the operator is mutated, what happens? If operator is mutated, there is no any access for binding a lac repressor protein. So all the time when there is a presence of lactose, all the genes or all the structured genes would get transcribed and translated. Lac repressor gene, if lac I gene is mutated or deleted, no any formation of a repressor protein, the operon would on all the time. Cap binding site, catabolic active protein binding site, if we delete this, there would be no any change on operon. Means these are the some scenarios, a summary. When a presence of glucose and lactose, no transcription occurs because glucose will be get depleted first, no cyclic AMP, the activator proteins not bound to a DNA, and it keeps following up a RNA polymerase from a promoter site, so no transcription. Glucose is there, lactose is absent. The activator protein would not bind to a DNA. The lac repressor protein would bind to a operator site, and no transcription would occur. No glucose, no lactose, no transcription. No glucose but lactose is there, a high level of transcription, high level of formation of the enzymes like beta galactosidase, permease and transacetylase. So thank you friends, thank you for watching me. Please subscribe my channel Easy Life Science, like my video, share it to your friends, and don't forget to press the bell icon so you'll get the notification of my newly uploaded video. Thank you once again.